I want you to, well, close your eyes for a second. Unless you're driving, obviously.
Yeah, please keep your eyes on the road if you're behind the wheel.
Exactly. But if you're safe at home, just picture a human lung for a minute. Usually we just see those two pink balloons from high school biology, right?
Right, the simple mechanical bellows that just inflate and deflate.
Yeah, exactly. But I want you to zoom in, past the tissue, past the air sacs, because if you look closely enough, the lung isn't a balloon at all. It's a sprawling, chaotic metropolis.
It really is. I mean, you have millions of residents to cells, you've got this intricate highway system of blood vessels, and you have the physical scaffolding of the buildings themselves.
And in a healthy city, everyone has a job. The trash collectors get the trash, the police patrol the streets, the builders fix the potholes.
But in chronic obstructive pulmonary disease, or COPD, that city is basically on fire.
And for decades, the way we've diagnosed this massive urban collapse is, honestly, it's kind of primitive. We just ask people to blow really hard into a plastic tube.
Right, spirometry.
Yeah, spirometry.
Which, it's useful, don't get me wrong. It tells us the traffic is jammed, but it tells us absolutely nothing about why.
Like, is there a riot? Is the bridge out?
Exactly. Treating COPD based just on airflow is like trying to fix a broken computer by measuring how hot the casing gets. You know it's failing, but you have no idea which microchip actually fried.
And that is exactly the mission of this deep dive. We are looking at a study that finally gives us the actual schematics of this broken city.
It's a game changer.
It really is. It's a massive paper published in Nature Genetics 2026. It's titled Aberrant Cellular Communities Underlying Disease Heterogeneity in Chronic Obstructive Pulmonary Disease.
It's a mouthful.
Yeah, very academic title. But when I say massive, I mean they looked at 1.5 million individual cells.
It's just an unprecedented data set. They used something called single-nucleus RNA sequencing on 141 study participants.
So they didn't just take a biopsy and look at it under a microscope to see if it looked inflamed?
No, not at all. They sequenced the genetic, think of them as the private diaries of every single cell type. They wanted to see exactly what those cells were thinking, what they were feeling, and what they were screaming out to their neighbors.
We are going to break this down, literally, cell by cell. We'll find out who the villains are, why the city's repair crews are failing, and how this might eventually give us a simple blood test to predict a patient's future.
It's a lot to cover.
It is, but we'll get through it. Let's start with the methodology, because you mentioned single-nucleus RNA sequencing. Why specifically the nucleus? Why not just sequence the whole cell?
That is a really great question, and it's basically the key that unlocked this entire study.
Okay.
Usually, if you want to sequence a cell, you need it fresh. It has to be alive. But if you want to study the deep history of a disease across hundreds of patients, you need to look at archived tissues.
You mean samples that have been sitting frozen in biobanks for years? Like the Lung Transplant Research Consortium?
Exactly, the LTRC. Now, when you freeze tissue, ice crystals form, and the outer cell membrane usually shatters. The cell basically bursts.
Oh, so the cell is dead and broken.
Right, but the nucleus, the little armored vault inside the cell where the DNA and the active RNA instructions are kept, that vault actually survives the freezing process.
That's incredible.
Yeah. So by sequencing just the nuclei, researchers could essentially travel back in time. They pulled out these frozen time capsules and read the gene expression profiles of 1.5 million cells from patients with all different stages of COPD.
And when they cracked open those files, they found a massive plot twist right away. I'm calling this the inflamed non-immune discovery.
That completely upends the dogma. I mean, for 50 years, we have thought of COPD inflammation as strictly an immune system problem.
Right. We blame the macrophages and the T cells. We blame the police force.
We thought, okay, the police are rioting, and that riot is destroying the city.
But this data says, well, it's not just the police.
No, it's the roads, it's the buildings, it's the drywall.
The actual structure itself.
Yes. They found that the structural cells, the endothelial cells that line the blood vessels, the epithelial cells that line the airways, and the fibroblasts that build the physical scaffolding, they were all stuck in this highly inflamed state.
Let's get really specific here because I want you listening to be able to visualize this. Let's start with the blood vessels. The study identified specific inflamed populations, right?
Right. They found specific subsets of endothelial cells. They labeled them arterial I and G-capi.
The I stands for inflamed.
Exactly. Arterial inflamed and general capillary inflamed. And these cells were actively expressing genes like NFKB1 and IRF1.
Okay, let's pause on NFKB1 because that sounds like a random license plate number, but it's really important. What does it actually do?
Think of NFKB1 as the master panic switch for inflammation inside a cell.
The nuclear fire switch.
I like that, yes.
Right.
When a cell flips that switch, it stops doing its normal job and starts pumping out distress signals. It releases inflammatory cytokines.
So here you have the lining of the blood vessel, which is just supposed to be a smooth pipe, letting blood flow, flipping the panic switch, and shouting for the immune system to attack.
Exactly. And they saw this in another critical cell type called the aerocyte. They found aerocyte I.
What's an aerocyte? It sounds like something from aviation.
They are these incredibly specialized, delicate capillary cells that physically wrap around the air sacs in your lungs to catch oxygen.
So they are the actual gas exchange interface.
Yes, and in COPD, instead of just catching oxygen, they are inflamed, stressed, and sending out alarm signals.
It wasn't just the plumbing that was acting up, though. The fibroblasts were going rogue, too.
Oh, absolutely. The researchers identified alveolar fibroblasti and adventitial fibroblasti.
So the scaffold of the lung is inflamed.
Right. And these fibroblasts were expressing those same NF-kappa B subunits, just like the blood vessels, but they were also churning out enzymes like 8-MTS4.
AMTS4, that sounds like a rogue demolition droid from Star Wars.
That is actually a perfect analogy. It's a protease. Basically, it's a molecular pair of scissors. Its only job is to cut up structural proteins.
So imagine a construction worker who just loses his mind and takes a chainsaw to the support beams of the house he's supposed to be building.
That is exactly what an inflamed fibroblast is doing.
So the infrastructure of the lung is actively participating in its own destruction. It's not a victim of the immune system. It's a willing accomplice.
That is the core takeaway. And they proved it. They didn't just look at the sequencing, they used immunofluorescence to show that these structural cells literally light up with inflammatory proteins right there in the patient's tissue.
Okay, let's do a quick recap so you don't lose anyone. It's been about 5 minutes. We're going to do these summaries throughout the deep dive.
Good idea.
Okay, recap bullet 1. The inflamed non-immune discovery. COPD isn't just immune cells rioting. The actual structure of the lung, the blood vessels, and the building scaffold are actively inflamed and destroying themselves, driven by that NFKB1 master switch.
Perfect. So that covers the what. Now we need to look at the when.
Right, section 2, the timeline of disease. Because the study shows that COPD isn't just a steady, straight line down, it's really a tragedy in 2 acts.
Act 1 is the attempted repair, and Act 2 is the collapse.
Let's start with Act 1, early disease. Maybe you're a smoker or you're exposed to harsh air pollution, your lung takes a hit. What actually happens?
The lung tries to be a hero. In early COPD, what we call Goldie stages, IND-ST-CO, the study found a massive surge in regenerative cell states.
The lung is trying to heal itself.
Yes. Specifically, they found a huge population of these stem-like repair cells called AT2S.
AT2S. And these had a specific marker, didn't they? YAP1.
Yes, the yes-associated protein. YAP1 is this powerful, universal signal for wound healing and cell proliferation.
I remember it as yes, always patching.
That's a great mnemonic. So in early disease, the lung's just flooding the zone with these AT2S repair crews. They're trying desperately to patch the holes in the airway lining.
But obviously, because people get severe COPD, it doesn't work permanently.
It fails miserably. As you move into Act 2, which is severe disease, Goldie stages 3 and 4, those repair crews just vanish. The AT2S cells crash and burn.
And nature hates a vacuum.
Exactly.
Right.
When the repair crews leave, the bad actors move in. The study highlights 2 specific villain cells that just take over the neighborhood.
The first one has a great acronym, the ABCs.
Aberrant basiloid cells.
Absolute bad squatters.
You could definitely call them that. In a healthy lung, these cells are practically non-existent. But in severe COPD, their numbers just explode.
What do they do?
Nothing useful. They are these weird dysfunctional cells that look a lot like the basal cells you'd find way up in your upper airway, but they migrate deep down into the fragile gas exchange area where they absolutely do not belong.
So they really are squatters.
Worse than squatters. They occupy the prime real estate where your body needs to absorb oxygen, but they can't exchange gas. They just sit there taking up space, forming these useless cysts.
Then the second villain that moves in.
The CTHRC1 positive fibroblast.
CTHRC1. Let's call them the collagen tissue-heavy remodeling crew. Because they make stars.
That's exactly what they do. Remember our road construction workers. These are a specialized crew that only knows how to do one single thing, lay down dense scar tissue. They are highly profibrotic.
So as the patient's ability to breathe drops, the number of these scarring fibroblasts skyrockets.
Exactly. So you lose your repair crew, the AT2S cells. And you get invaded by squatters, the ABCs, and scar makers, the CTHRC1 fibroblasts.
And meanwhile, what is happening to the good guys, the delicate cells that actually let you breathe?
They're physically wiped out. The study showed a catastrophic depletion of those aerocytes we talked about in AT1 cells.
AT1 cells being the ultra-thin cells that physically allow oxygen to slip into your bloodstream.
Right, they die off, which is exactly why severe COPD patients feel like they're constantly suffocating.
It's not just that the airways are blocked so air can't get in, it's that the actual cellular machinery required to process the air is completely gone.
Precisely. The city's oxygen factories have been bulldozed.
Okay, let's lock this in with recap 2.
Go for it.
Bullet 2. The 2 acts of COPD. Early disease is a valiant attempt to heal, marked by AT2S cells and the YAP1 repair signal. Late disease is a total failure of repair.
And that failure leads to an invasion of aberrant basiloid cells, the squatters, and CTHRC1 fibroblasts, the scar makers, while the crucial gas exchange cells just die off.
Which brings us to a massive question. How did these cells organize themselves? Which leads to section 3, cellular communities.
The bad neighborhood.
Right, because cells don't just float around in a vacuum, they live next to each other.
And your neighbors dictate your behavior.
Yeah.
To figure out who lived where, the researchers used a cutting-edge technology called Xenium.
Xenium sounds like a sci-fi planet.
It's actually a form of spatial transcriptomics.
Which means?
Well, imagine taking a high-resolution satellite photo of our lung city. Traditional sequencing tells you, hey, there are 500 criminals somewhere in this city. Xenium spatial transcriptomics tells you all 500 of those criminals are living exactly on 5th Street, right next to the bank. It maps the gene expression in physical 2D space.
That is wild. And by looking at the map, they found 2 distinct, dangerous neighborhoods that basically determine a patient's fate.
Community 5 and Community 1.
Let's dig into these, because this is the bridge to actual patient symptoms. Let's start with Community 5.
Community 5 is the high inflammatory neighborhood. This is the combat zone. All those inflamed structural cells we talked about earlier, the AT2I epithelial cells, the GCAPI inflamed capillaries, they all physically cluster together here.
So they are throwing a massive inflammatory block party.
Basically, yes.
Yeah.
And the clinical link is what's truly fascinating. When they looked at patients whose lungs were dominated by Community 5.
What did those patients look like in the clinic?
They were the highly symptomatic phenotype. These are the miserable patients. They have a huge symptom burden, constant hacking coughs, massive phlegm production.
They're wet and inflamed.
Yes. And they suffer from frequent exacerbations, those terrifying flare-ups where they suddenly can't breathe at all and end up rushed to the ER.
So Community 5 equals active hot fire. Now, contrast that with the other bad neighborhood, Community 1.
Community 1 is the fibrotic neighborhood.
The scar zone.
Right. This zone is completely dominated by those CTHRC1 positive scarring fibroblasts.
And how does that patient look in the clinic?
This is the crazy part. They might blow the exact same numbers on a spirometry test as the Community 5 patient.
But their experience of the disease is totally different.
Completely different. They might not cough much at all. They aren't producing phlegm. They are dry.
But they still can't breathe.
Because their lungs are turning into rigid, stiff scar tissue. They suffer from severe dyspnea, shortness of breath, but that active hot inflammatory fire might actually be completely burned out.
This is a huge aha moment for anyone listening.
Oh.
We have spent decades treating both of these patients as if they have the exact same disease, just giving them the same standard inhalers.
Exactly. But one patient has a raging fire in their chest, and the other patient is sitting in a burnt-down, scarred house. Mix them together in a clinical trial, and of course your new drug is going to fail.
Now, we can't talk about neighborhoods without talking about the police. The immune system is still there.
It is. And in these bad neighborhoods, there is a very specific enforcer, a macrophage with the designation IMCHIT1.
IMCHIT1, interstitial macrophage expressing kinase 1. I remember these as the riot police who show up to a protest and just start breaking store windows.
Very accurate analogy. These are heavily profibrotic macrophages. They express destructive genes like MMP9 and SPP1.
And they love hanging out in those remodeling neighborhoods.
They do. But here's the most terrifying thing the study found. The lung actually starts building dedicated military bases just to support these rogue cops.
The FRCs, fibroblast reticular cells.
Yes. Normally, you only find FRCs inside your lymph nodes, which are the immune system's command centers.
Right.
But in severe COPD, these FRCs start appearing deep inside the actual lung tissue. They build what are called tertiary lymphoid structures.
The lung is literally militarizing itself. It's building forward operating bases right on the delicate air sacs to pump out more inflammation. It's horrifying, but brilliant.
Which brings us to section 4, the mechanism. How are all these cells coordinating this?
They looked at cell communication, specifically ligand-receptor interactions.
Let's translate that. A ligand is basically a text message sent by one cell, and the receptor is the specific cell phone app on another cell that can receive it.
Exactly. And they found this vicious repeating cycle.
Who sends the first text?
The structural cells. The damaged, inflamed blood vessels send out signals like CXCL10 and CXCL11. The fibroblasts send out CCL2 and IL-33.
Basically dialing 911. Help, we are damaged.
Right. And who picks up the phone? Our rogue riot police. The IMCHIT1 macrophages have specific receptors like CXCR3 that hear that exact call. So they rush out of the blood and into the lung tissue.
But when they arrive, they don't help fix the pipes. No. They release massive amounts of proteases, specifically one called MMP12.
And MMP12 eats elastin.
Right. Elastin is the rubber band protein that makes your lungs stretchy so they can recoil when you exhale. The macrophages chew it all up.
So let's trace the loop. Structure gets damaged and calls for help. The macrophage arrives. The macrophage eats the structural elastin. The structure gets more damaged and screams even louder.
And the cycle repeats. Forever.
Wow. The paper also did this really interesting mediation analysis regarding smoking, because we all know smoking causes COPD, but this paper connects the dots on exactly how.
It connects the who to the what, they asked. Does cigarette smoke directly make you short of breath? Or does the smoke trigger a middleman?
And the middleman is our riot cop, IMCHIT1.
Exactly. The statistical analysis showed that the effect of smoking on dyspnea, the actual breathlessness, is directly mediated by the expansion of those specific macrophages.
Smoking recruits them, and then they cause the symptom by destroying the tissue.
Conversely, having healthy AT1 cells, those delicate gas exchange cells, mediates protection against breathlessness.
So if you could somehow invent a drug to block just the IMCHIT1 macrophages, maybe you could break the link between smoking and the worst lung damage?
That's the dream.
Okay, let's do recap 3.
Bullet 3. Cells form spatial neighborhoods. Community 5 is high inflammation and drives severe symptoms like coughing. Community 1 is high scarring and drives stiffness.
And a vicious cycle sustains this. Inflamed structure calls in the IMCHIT1 macrophages, which destroy the elastin with MMP12, causing more structural panic.
It's a self-feeding loop.
But here is the massive clinical problem. I can't take a core biopsy of every single COPD patient's lung just to see if they live in Community 5 or Community 1. It's too dangerous.
You'd collapse their lung.
Exactly. So we need a window from the outside. Which brings us to section 5, the liquid biopsy.
This is the immediate practical application of this paper. The researchers looked at plasma proteomics. They took simple blood samples from the patients and screened a panel of 5,400 different proteins using Olink technology.
They were hunting for a mirror, a reflection in the blood of what was happening deep in the lung tissue.
And they found it. They used unsupervised machine learning to cluster the blood proteins, and they identified a specific group they called Module ME2.
Module ME2, the mirror environment. What exactly is an ME2?
It's a potent cocktail of inflammatory markers. You've got TNF signaling proteins, IL-17 signals, and proteins that regulate that master NF-kappa B switch we talked about earlier.
And here's the absolute kicker. If a patient has high levels of module ME2 floating in their blood,
it almost perfectly correlates with them having the high inflammatory community 5 physically dominating their lung tissue.
That is huge. That is literally a simple blood test for internal lung architecture.
It allows us to finally stratify patients. Right now, like we said, everyone gets the same steroid and bronchodilator inhalers. But with this, we could take a blood draw in the clinic and say, okay, you are ME2 high, you have inflammatory COPD.
You need drugs that target cytokines.
And you are ME2 low. You have fibrotic COPD. Steroids won't do anything for you.
It's precision medicine, the holy grail for pulmonology.
It really is.
We're in the home stretch here, so let's synthesize all of this with a practical case study.
Let's play doctor.
All right, scenario. 2 patients walk into your clinic, patient A and patient B. You give them both a spirometry test, and they both have exactly 50% of normal lung function.
On paper, they look identical.
But patient A is ME2 high on our new blood test, and they complain of a chronic wet cough.
Based on this deep dive, we know patient A has community 5 disease. Their structural blood vessels are actively screaming. Their NF-kappa B switch is taped down. So how do we treat them?
Well, the study mentioned specific targets like IL-4R and TNF.
Right, so we might borrow highly effective drugs from asthma or autoimmune diseases. Biologics like dupilumab, which blocks IL-4R, or maybe a TNF inhibitor. We need to pour water on the fire in the endothelium.
Now look at patient B. They test Me2 low. They are completely dry, no cough, but they are gasping for air when they walk up a single flight of stairs.
They have community 1 disease. The fire is mostly out, but the scar is permanent. Anti-inflammatory biologics would be useless for them.
But what do they get?
They might need anti-fibrotic drugs. Things like nintedanib, which we currently use for a totally different disease called idiopathic pulmonary fibrosis.
If we treated patient B with patient A's anti-inflammatory drugs, we'd just be wasting time, wasting money, and letting their disease progress.
And frankly, that is exactly what the medical field has been doing for 30 years. That's why so many COPD clinical trials fail. We average out the patients instead of treating the individual biology.
This paper fundamentally rewrites the definition of the disease. It moves us from COPD is a smoker's disease to COPD is a failure of regeneration and a specific spatial organization of aberrant cells.
It maps the chaos.
Before we sign off, I want to leave you, the listener, with a slightly provocative thought to chew on.
I love a good paradigm shift.
We spent a lot of time today talking about the arterial eye and G-Kappa cells, the inflamed blood vessels.
Right, the plumbing.
If the structural blood vessels are driving the inflammation just as much as the immune cells, why do we strictly treat COPD as a lung airway disease? What happens if we start treating it as a vascular disease?
That is the ultimate frontier. Right now, we have patients inhale drugs to try and reach the airway lining. But if the actual distress signal is coming from the capillary blood vessels,
maybe we need systemic intravenous treatments.
maybe we need to heal the blood vessels first in order to save the air sacs. If you fix the city's plumbing, maybe the whole neighborhood finally recovers.
From just blowing in a plastic tube to healing the city streets from the inside out.
It's a brave new world for respiratory medicine.
It really is. If you want to dive deeper into this, we have linked the Nature Genetics paper in the show notes. I highly, highly recommend clicking through and looking at the visual maps of Community 5 versus Community 1. It makes the invisible totally visible.
It's absolutely worth the click.
Thanks for joining us on this deep dive. Stay curious.
Stay informed.